DNA cycle sequencing is also known as the Sanger method, but it's a method used to determine the order of the nucleotide bases in a strand of DNA. So it determines adenine, guanine, cytosine, or thymine. And it starts out very similar to PCR in the way that it, first step is denaturation. So you heat the strands, separate them, and there it changes. So then you only use one primer in cycle sequencing, and you only end up with one strand instead of the two. So another difference is that along with the nucleotides, you're also adding dideoxynucleotides, and these are changed nucleotides that they're also called terminating bases because they have a hydrogen group at the end instead of a hydroxide group. So whenever the DNA strand is elongating, it can't bond beyond this because the hydrogen, it needs to bond to hydroxide and it can't because there's only hydrogen. So it stops the replication. And because it's only a single strand, you end up with linear amplification, whereas with PCR, it was exponential because it was doubling each time we started the cycle. So that means that any future copies or applications you need to make, you must make them from the original template. You can't, just keep going like you can with PCR. So first, it starts with denaturation. So you heat the strands, and you have 4 test tubes now instead of just the one, because you're determining each of the bases. And you denature them so they separate, and then you add the primer, and that will form the new strand, and then the dideoxynucleotide will stop the synthesis. So then you take each of your 4 test tubes and you place them in polyacrylamide gel, and then you apply it to gel electrophoresis, where an electrical current is applied, and where the DNA is, it's a negative current, and DNA is also negatively charged, so it's going to travel down through the gel to get to the positive end, because that's where it's attracted to. And any smaller strands of DNA are going to travel a lot faster than the larger strands, so it'll get down towards the end faster. And the dideoxynucleotides, they also have a fluorescent tag on the ends, so whenever you do gel electrophoresis, the color lights up as these dashes. So whenever you look at your results later, you can see the order that they go in. And so you look at it's an electrophoregram, and it shows the colored peaks of each of the nucleotides, and you read it from bottom to the top, so the smallest strands to the largest strands, and the color will coordinate to whichever base it was. So like for example on this one, like the smallest strand matches up with thymine, so then it goes T, then cytosine, and so on.